How to put it on the other screen? Sorry, yeah, I'm trying to use this. Yeah. Hi everyone, my name is Javi. Today I'm going to be introducing to you a web tool called PopNetD3 for the analysis and visualization of population structure. Okay, so in the past few years, our access to whole genome sequences has increased significantly. In the NCBI database alone, the number of sequenced genomes has increased from 18,000 from 2013 to over 100,000 in 2017. And this increased availability in data has really enabled population scale studies, for which an understanding of population structure is very crucial. Now, population structure refers to the composition of a population and can answer questions like how many subpopulations we have, which sample belongs to which subpopulation, and more importantly, whether any relationships exist between either the subpopulations or the individuals in your population. So in this example, a study on Toxoplasma gondii involving 138 samples from all over the world found 6 clades, as shown here, using this neighbornet sort of visualization. And they were able to find evidence of admixture and recombination between the clades. So we thought the discovery of admixture between clades was really interesting and important. And so last year we published a tool called PopNet that focuses specifically on this. So PopNet represents a population using a network as opposed to a tree like you saw earlier, and uses chromosome painting to identify these recombination events. So very briefly, the PopNet algorithm starts with a table of whole genome SNPs for all of your samples. Then we divide up this table into sections, each corresponding to one specific section of the genome. So like chromosome 1, base pair 1 to 50 or something. And we create a similarity matrix using the SNPs in each section, and we cluster it using Markov clustering to create a series of clustering patterns, with the idea that when a recombination event occurs, it'll affect the clustering pattern within a few of the sections, allowing you to identify it. Now, in the next step, we aggregate all the co-clustering or pairwise co-clustering frequencies, which is how frequently 2 samples occur within the same cluster over all the sections. And we use that to create a new similarity matrix, which we then cluster again to define our subpopulations, like other programs would. And we assign a unique color to each of the subpopulations we find to use in our chromosome painting. So in a chromosome painting, for each sample, we have 1 bar to represent its genome, and we color it according to who we think it's most similar to at each section, or which subpopulation we think is most similar to at each section. And we embed these chromosome paintings into nodes of a network to produce the final visualization. So after publishing PopNet, we thought about how to make it more accessible and user-friendly, and arrived at the second iteration called PopNetD3, which transforms PopNet into a web-based platform. So to use it, just go to our website, comsysbio.org/PopNetD3, and go to the job submission portal, where you fill in a simple form with 8 fields, and 3 parameters, upload a file, and hit the submit button to run PopNet on our server. Now, our requirement for the file you submit is also pretty simple. We can only handle haploid genomes right now, and we're expecting whole genome SNPs for all your samples in this tabular format that looks like this. So we have a header row, and every row is genotype at 1 position. And there will be a full description of this on the website. And when your job is done, you'll be notified by email and can view your network in our new D3-based, hence a D3, genome or network visualizer. So in this example here, I have 20 Toxoplasma gondii genomes from North America. Each node here is one genome, one sample, and edges, or the edge weights, represent the overall similarity between the connected nodes. So thicker means more similar overall. And the first observation you can make about this network is that there are 4 subpopulations, as indicated by the 4 different border colors: green, blue, pink, and yellow, and I'll be referring to them as such. So normally we would expect some strong edges or thick edges between members of the same subpopulation, but the interesting ones are usually the links between members of different subpopulations, which could indicate the presence of a hybrid or ancestral relationships between subpopulations. And looking inside each node, we can see a circular chromosome painting. Now the pattern is pretty complicated, but what it allows you to do is quickly compare 2 samples and see where they're the same and where they're different. So comparing B73 and ME49 from the blue subpopulation, you can see that most of their genomes are pretty similar, except for this part here, where B73 is similar to the pink subpopulation. And so this suggests that B73 could be a hybrid between something that looks like ME49 and a member of the pink subpopulation. And so the network here gives you an overview of this chromosome painting, but you can also access the detailed version by selecting a particular chromosome and clicking this button on the network visualizer. And I'm going to give an example of this on the next slide using chromosome 4, which is this yellow region over here. So this is chromosome 4 of 4 of the blue subpopulation members, B73, ME49 from the previous slide, as well as PRU and GOAT US21, which are these 2. Now from the network, it seemed like they were all similar to the yellow subpopulations throughout the entire chromosome. But upon closer examination, you can see that quite a few sites have in fact diverged from the yellow subpopulation, except in PRU, where these blue sites are actually missing. And this is most likely due to additional, more recent recombination events between PRU and the yellow subpopulation. Yeah, and if you had phenotype data suggesting that PRU behaves differently than the other 3, then this would be an interesting place to look. And all the data I've shown in this presentation are available on the website as an example, where you can go and kind of play around with the network yourself. Also, you can come to my poster later on this evening for a live demo where I'll use it on my tablet. And with that, I'd like to thank my supervisor, Dr. John Parkinson, the rest of the Parkinson lab, the organizers, and everyone here. Thank you.